Hello. I've got one more class of molecule to talk to you about, the nucleic acids. This presentation is a little bit shorter than the other ones, and that's because when we get into genetics later in the semester, we're going to talk into a lot more detail about nucleic acids. So right now it's just a brief introduction to those. So for what do we use nucleic acids? There's really two types, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Know that. Your chromosomes are all made of DNA. And so a long strand of DNA, and then with some proteins wrapped around it, that is what a chromosome is. The DNA carries instructions for making RNA. So one nucleic acid has the instructions for making another nucleic acid. And then the RNA then carries instructions for making proteins. So again, remember our mantra. Before you go to bed at night, DNA makes RNA, makes protein. Right? We're going to talk about the details of how one makes the other when we talk about genetics. Okay, so what are the building blocks of nucleic acids? The building blocks, the monomers, are nucleotides. Now each nucleotide has three parts to it: a nitrogenous base, a pentose sugar, and a phosphate group. And so here on the left, you're looking at a string of these nucleotides stuck together. And then on the right, we zoom in on one of those. So you see the base, the nitrogenous base is at one end, and it's attached to a pentose sugar, so a 5-carbon sugar. So again, count the vertices, and you see there are 5 carbons. And then on the 5' carbon, you've got a phosphate group. Remember that a phosphate is a phosphorus with 4 oxygens attached to it. It's a very reactive molecule. So all nucleotides have this basic structure. Now, there's only 5 different nitrogenous bases. And we typically use their abbreviations: C, T, or U. We'll talk about that in a second. A and G. It stands for cytosine, thymine, uracil, adenine, and guanine. Okay? Now, if you look at the pentose sugars, there's only 2 types of pentose sugar: deoxyribose and ribose. Okay? And that's where the name DNA and RNA get their name from. Now, look at the ribose sugar that's on this slide. Okay? And you see in the lower right carbon, you've got a hydroxyl group in red, that OH in red. Now, look at the same carbon on the deoxyribose. You see that oxygen is missing. Ribose is a sugar. If you take away an oxygen or you deoxy it, you get deoxyribose. That's where the names come from. All right? And so from those 5 types of nucleotides, we can build any DNA or RNA molecule that we want. There's really only 4 types. Again, thymine and uracil are interchangeable. One is used in DNA and one is used in RNA, right? So what are some differences between DNA and RNA? DNA is double-stranded. RNA only has 1 strand. DNA uses deoxyribose as its pentose sugar. RNA just uses ribose. DNA only uses C, G, A, and T: cytosine, guanine, adenine, and thymine. Those are the 4 types of nucleotides that are used to build DNA. And those are, the nitrogenous bases are what give them their name. RNA uses C, G, A, and U. And so that's just a difference between the 2 types of molecules, right? So uracil is used in RNA, thymine is used in DNA. So again, the DNA makes the RNA, which makes the protein. So nucleic acids, your DNA or your RNA, are long, unbranched strands of nucleotides. So remember we said that about proteins, right? Proteins are long, unbranched strands of amino acids. So it makes sense that they both have the same basic shape. Since DNA makes RNA, makes protein, they're all just a long chain of monomers, right? They don't have pieces going off. And so you have a DNA strand, and then that will make an RNA strand, and then that will make an amino acid strand, and then that amino acid will fold into the characteristic shape of the protein. The order of nucleotides in the DNA determines the order of nucleotides in the RNA. The order of nucleotides in the RNA determines the order of amino acids in a protein. And so that's why we say DNA makes RNA makes protein. So it all starts with the DNA. Whatever order those nucleotides are in determines the order of the nucleotides for the RNA, which determines the order of the amino acids in the final protein. So if you change the DNA, if you change, if you swap out a base or you move a piece over, then you're going to change the order of nucleotides in the RNA. And if you change the order of nucleotides in the RNA, that might change the order of amino acids. When we get into genetics, we'll see why, it doesn't guarantee that it's going to change the amino acids, but it might change the amino acids. And that's what a mutation is, right? A mutation is a change in the DNA. And any mutation in the DNA will cause a mutation and a change in the RNA, but it only might change the protein. Okay, and so with all the other groups, carbohydrates and lipids and proteins, you're familiar with those terms from your diet nutrition. You don't hear about nucleic acids very much. When we talk about, you got to eat your DNA, right? You don't really hear that very much. Well, what foods are high in nucleic acids? They all are, right? Every cell has DNA. Every cell has RNA. So no matter what you're eating, it has nucleic acids. And so it's just something that it's never a significant factor in your diet. You always have plenty of nucleotides to build your DNA and RNA. All right. I told you this one was short, but we're going to get into a lot more detail about the nucleic acids when we talk about genetics later in the semester. So that's it, and I will talk to you later.